Welcome back, everyone, for another deep dive. This time we're going microscopic into the world of gram-positive anaerobic Cocci, GPAC.
You got it.
And for this, we're taking a deep dive into Principles and Practice of Clinical Bacteriology, second edition. You know these bacteria. They're often overlooked, but they're clinically significant, especially for all you medical microbiologists out there.
That's right. They're often dismissed as just commensals, but GPAC can cause a whole bunch of infections, and they can be tough to diagnose and treat.
And there's been so much new research that's really changing the way we classify and understand these organisms.
Absolutely. It's an exciting field that's rapidly changing.
So let's just jump right in, start with the basics.
Yeah.
You might remember the term anaerobic streptococci from back in the day in your micro textbooks.
Oh, yeah. But that term's outdated and actually pretty misleading. We know now that these organisms aren't actually closely related to streptococci at all.
Okay.
They're on a totally different branch of the bacterial family tree, mostly within the Clostridiales order.
So, ditch the old terminology. Let's get into who the major players are using their updated names. Who should we be looking out for in the lab?
There are a few big culprits that pop up all the time in those clinical specimens. Clinigoldia magna, for example.
Okay.
That one's a real pathogen, often found in soft tissue infections, bone and joint infections, even diabetic foot ulcers.
Diabetic foot ulcers. That's a serious clinical connection.
Absolutely. You know, it highlights that these GPAC aren't just causing those minor skin infections. They can be involved in some really complex cases. And another common one is Micromonas micros.
Okay.
Now, this species, it loves to hang out in the oral cavity, so you'll find it in dental infections and complications from those.
Makes sense. What else should we know about Micromonas micros?
Well, it's known for producing a lot of hydrogen sulfide,
Okay.
which is a cytotoxic compound, so that probably contributes to its ability to cause all that damage in the oral cavity.
Interesting. What about Piptoniphelus and Saccharolyticus? That's a mouthful.
It is a mouthful. This one's kind of a trickster. It's common, but it often gets misidentified, which can lead to, problems with treatment. So it's really important to be aware of the characteristics and use really accurate identification methods.
Got to be extra careful with this one. Any other GPAC species that we should be familiar with?
Anaerococcus vaginalis. As the name suggests, it's often found in the vaginal flora, can be associated with a whole range of gynecological infections.
Okay, starting to get the picture here. And then there are a few less common but still relevant genera.
Exactly. Peptostreptococcus, ruminococcus, coprococcus, and sarcina. Those can also show up occasionally, so it's good to have them on your radar.
Got it. Now let's talk about their, how they live. These GPAC are anaerobes, but some can tolerate oxygen for a while, right?
Right.
How does that affect what we do in the lab?
Oh, that's a crucial point. The fact that they can survive with oxygen around for a bit means we need to be super careful about how we collect and transport specimens.
The speed is key.
Absolutely. You got to use the appropriate anaerobic transport systems and get those samples to the lab quickly. Otherwise, you risk missing these organisms.
Okay, good to know. Now let's dive into how they cause infections. How do these GPAC actually do it?
One of their main strategies is microbial synergy.
Okay.
They often team up with other bacteria to create a much stronger infectious force. It's like a multi-pronged attack.
So it's not always a solo mission.
Right, exactly. This power in numbers approach can make GPAC infections really tough to treat, but they also have their own individual weapons.
Like what kind of virulence factors are we talking about?
Well, for example, Clinigoldia magna. It produces proteins that bind to human serum albumin and immunoglobulins that can potentially mess with the immune system.
So they're not just attacking directly, but also trying to weaken the host's defenses.
Exactly. And some GPAC, like Micromonas micros, produce capsules.
Capsules.
Yeah. Capsules can make bacteria more resistant to phagocytosis, which is, a major way our immune system destroys invaders. So it's like they're wearing armor.
Exactly. And these capsules can make GPAC infections stick around longer and much harder to clear.
Wow. They're more sophisticated than I thought. All right, let's switch gears and talk about what this looks like in the clinic. What kind of infections are we actually seeing with GPAC?
Well, they're actually involved in a surprisingly wide range of infections, and their role is often really underestimated. They're actually the second most common anaerobic bad guys, responsible for about 25 to 30% of anaerobic infections.
That's a lot. I had no idea they were that common.
Yeah, and they can affect all sorts of different parts of the body. We already talked about diabetic foot ulcers, but you find them in oral and respiratory tract infections, intra-abdominal infections, genitourinary infections, skin and soft tissue infections, even musculoskeletal infections.
Well, they're not picky about where they set up shop.
Not at all. They can cause everything from minor abscesses to serious, life-threatening conditions. This is really making me rethink how I think about GPAC. Okay, so we know they cause a lot of different infections, but how do we even diagnose them? What are the key things to look for in the lab?
Well, those traditional biochemical tests, they're not always reliable for GPAC.
Okay.
They can take a long time, and they often lack the specificity that we really need.
So what's the solution? How do we accurately identify these bacteria?
Thankfully, we have these preformed enzyme profile kits now, PEP kits. They're much more accurate and efficient.
PP kits. Tell me more about those.
Yeah. These kits, they have a panel of preformed enzymes that react with very specific substrates. They produce a unique color pattern for each species. It's like a fingerprint.
Oh, wow. That sounds really useful.
Mhm.
Any tips for using these PP kits effectively?
Yeah, the key is to use a heavy inoculum to make sure there are enough bacteria to react with the enzymes in the kit.
Okay.
And always, always pay attention to cell and colonial morphology. Combining those classic observations with the PP results, that can really solidify your identification.
Great advice. So PEP kits are our current go-to. Any exciting new advancements coming down the pipeline for diagnosing GPAC?
Absolutely. Nucleic acid-based techniques are becoming more and more important. Things like PCR and other molecular methods, they offer even more precision and can detect GPAC directly from clinical specimens.
So it's a combination of classic techniques and cutting-edge technology. That's fascinating. Now, let's zoom out a little bit, talk about where these organisms typically hang out and how they spread.
Well, here's the thing. GPAC are masters of disguise. They're part of the normal flora on a lot of body surfaces.
Right. We mentioned that earlier.
So it makes it really hard to tell who's a friend and who's a foe.
Right. So they're already there, lurking in the shadows, waiting for an opportunity to strike.
Exactly. And this makes prevention really tricky. You can't just wipe them out from the body.
Right.
Because they're part of the normal ecosystem.
Okay. So if we can't eliminate them, how do we stop them from causing infections?
Well, prevention really focuses on maintaining that balance of the normal flora and minimizing the chances for GPAC to invade. So things like practicing good hygiene, proper wound care, and being mindful of things that can disrupt that microbial balance. Things like antibiotic use.
So keeping them in check.
Exactly. It's a balancing act.
Got it. Now, what about treatment? What are our weapons of choice when these GPAC decide to go rogue?
There's this common misconception that all GPAC are universally sensitive to antibiotics. But that's not true.
Okay, I'm all ears. Let's debunk that myth.
Different GPAC species, they can have very different susceptibility patterns. What works well for one might not work at all for another. So relying on broad-spectrum antibiotics without knowing exactly what species you're dealing with, it's like shooting in the dark.
So accurate identification is really important for targeted treatment.
Absolutely. It's essential. Now, metronidazole, that's usually a good first choice for a lot of GPAC infections. It's got excellent anaerobic coverage.
How about penicillins?
Penicillins can work, but resistance, especially in Streptococcus anaerobius, can be an issue.
So we need to be aware of that potential resistance. What are some other options?
Well, clindamycin can be effective against some GPAC, but resistance to that one is highly variable. Always a good idea to check susceptibility patterns before using clindamycin.
Can't just assume it'll work.
Right. And then finally, cephalosporins and carbapenems. They're usually reliable options for GPAC infections.
Okay, so we've got some choices, but species-level ID is key.
Couldn't have said it better myself.
Now, besides antibiotics, what else should we be thinking about when it comes to managing these infections?
Well, antibiotics are really important, but they're not always enough on their own.
Right.
We also need to think about draining abscesses, debriding infected tissue, and addressing any underlying conditions that might be contributing to the infection.
So it's a holistic approach.
Exactly. It's about treating the patient, not just the infection.
Makes sense. I think we've covered a lot of ground here. Time to put your knowledge to the test with a little quiz. Ready to see how much you've learned?
Bring it on. Feeling good.
Okay, first question. True or false? All GPAC are universally sensitive to metronidazole.
False. Metronidazole is a good first-line option for a lot of GPAC infections, but we do see resistance pop up. We always need to think about those species-specific susceptibility patterns. And ideally, we should do susceptibility testing to help guide our treatment decisions.
Okay, good point. We can't make assumptions about antibiotic sensitivity. Question number 2. Which GPAC species is particularly notorious for producing hydrogen sulfide?
That would be Micromonas micros. This species, remember, it loves the oral cavity and that hydrogen sulfide production. Well, that probably plays a role in how it causes problems there.
Interesting. Okay, question 3. A patient comes in with a suspected anaerobic infection. The gram stain shows gram-positive Cocci in clusters. What can you conclude, and what would your next steps be for identification?
Gram-positive Cocci in clusters. Well, that definitely raises suspicion for GPAC, but we can't just jump to conclusions.
Right.
Other organisms like Staphylococci, they can look like that too. So to really nail down the culprit, we got to think about where the specimen came from, the clinical presentation, and most importantly, do more testing. A p-pit would be a great next step, along with looking at colonial morphology and other characteristics.
So it's all about putting those classic microbiological observations together with the newer techniques to get a definitive ID. All right, question 4. Which GPAC species is commonly associated with diabetic foot infections and is often recovered in pure culture from those infections?
That would have to be Filamentous magna. This species is a true pathogen. It can cause infections all on its own, and it really seems to like those diabetic foot ulcers, so it's definitely something to keep in mind in those cases.
Okay, last question. Why is getting to the species level so important when dealing with GPAC infections, especially for choosing the right antibiotic treatment?
Well, as we've talked about, different GPAC species can react very differently to antibiotics. So using a broad-spectrum antibiotic without knowing exactly which species you're dealing with is kind of like using a shotgun.
Okay.
You might hit something, but you also risk a lot of collateral damage. Species-level ID lets us get more specific, choose the best antibiotic, and hopefully minimize the risk of antibiotic resistance.
Excellent point. It really highlights how important the role of the medical microbiologist is in guiding treatment decisions and really promoting good antibiotic stewardship.
Absolutely. Our expertise is essential to make sure patients are getting the most appropriate therapy.
All right. Now that we've tested your GPAC knowledge, let's dig a little deeper into some of the specifics of these organisms.
Okay.
Especially those details that are most relevant to medical microbiologists like you. We'll start with Streptococcus anaerobius.
Ah, yes. This species is interesting because it's the only one left in the genus Streptococcus. And get this, based on genetic analysis, it's actually more closely related to certain Clostridium species than to other GPAC.
So it's a bit of an outlier.
You could say that. It's got some unique features that set it apart. For example, it has this very distinctive, I'd say almost sickly sweet smell.
Interesting.
And it's sensitive to sodium polyethyl sulfonate, SPS, unlike most other GPAC.
Okay.
The sensitivity to SPS actually, it can be a helpful clue when you're trying to identify it in the lab.
Interesting. So a good sense of smell can come in handy in microbiology. Besides the smell and the SPS sensitivity, what else should we know about Streptococcus anaerobius?
Well, clinically, it's important to remember that it's often found in GI and high vaginal infections, but usually as part of a mixed infection with other bacteria. It's not always clear what its exact role is in these infections, but it's important to be aware of it, especially since some strains can be resistant to certain beta-lactam antibiotics.
Okay.
And that resistance, we think it's due to modified penicillin-binding proteins.
So it's one to keep an eye on, especially in those mixed infections. Now let's talk about the star of the show, Filamentous magna. We've mentioned it a few times already, but let's really break down its key features and clinical significance.
Okay. Filamentous magna is a fascinating and formidable species. I mean, it can cause a huge range of infections, from minor skin infections to really serious life-threatening conditions like endocarditis or brain abscesses. It's often found in soft tissue infections, bone and joint infections, and as we said before, diabetic foot ulcers. What makes it so successful is its arsenal of virulence factors.
Tell me more about those virulence factors. What makes it so good at causing infections?
Well, it produces a lot of different enzymes, including collagenase and jaldanase. These allow it to break down tissues and spread. It can also bind to human serum albumin and immunoglobulins, which may help it evade the immune system. And some strains produce a toxin called peptinylation F that can actually damage host cells.
Wow, it's a real threat. No wonder it can cause such serious infections. How do we identify it in the lab?
Cell and colonial morphology can give you some hints, but for a definitive ID, PEP kits are really the most reliable method.
And what's the good news when it comes to treatment?
The good news is that it's generally susceptible to metronidazole and beta-lactam antibiotics. However, there are reports of resistance to clindamycin, so it's always a good idea to do susceptibility testing.
Okay, good to know. Let's move on to another significant player, Micromonas micros. What are the key things to remember about this species?
Micromonas micros. Well, it's a major part of the oral flora, often found in dental infections, things like periodontitis and peritonsillar abscesses, and it's known for producing a lot of hydrogen sulfide from glutathione. That probably contributes to tissue damage in the mouth.
So it's a master of oral mayhem. Anything else about its behavior?
Well, it's often found in these mixed infections with other oral bacteria, those classic polymicrobial abscesses. And interestingly, there are two main morphotypes. A smooth one and a rough one. The rough morphotype is actually more often associated with infections, and it tends to be more resistant to antibiotics, so that's the one we really need to be aware of in the clinic.
Two faces of Micromonas. That's fascinating. How do we tell the two morphotypes apart?
They have different colonial morphologies, and you can also use genetic and electrophoretic techniques to distinguish them.
And what about treatment? How do we tackle Micromonas micros infections?
It's usually susceptible to most antibiotics, but there have been some reports of metronidazole and cefoxitin resistance, especially in strains from periodontitis. So susceptibility testing, once again, is really key.
Got it. So we can't get complacent about resistance, even with those commonly used antibiotics. Okay, let's move on to Peptinophilus saccharolyticus. What makes this species stand out, and what are the challenges in identifying it accurately?
Peptinophilus saccharolyticus. Well, it's another one of those common GPAC species, but it can be tough to identify. It often gets grouped together with other closely related species like Peptinophilus indolicus and Peptinophilus hooray.
So mistaken identity.
Exactly, and that can be a problem clinically because these species might have different susceptibility patterns.
Makes sense. We need to know who we're dealing with to pick the right antibiotic. So how do we differentiate Peptinophilus saccharolyticus from its close relatives?
It's asaccharilytic, so that means it doesn't ferment sugars, and it makes butyric acid as its main volatile fatty acid.
Okay.
However, those characteristics are shared by its close relatives, so those PEP kits aren't always reliable for distinguishing between them. We have to use those more subtle clues like cell and colonial morphology to really get a definite ID.
So it's a real test of our microbiology skills.
Mm-hmm.
What about the clinical significance of Peptinophilus saccharolyticus? Where do we typically see it causing problems?
It's part of the normal flora in the gut and genitourinary tract, and it's been found in infections at both sites. We don't have a ton of data on its pathogenicity yet, but it looks like capsule formation might be an important virulence factor.
Okay, so it's one to keep an eye on as we learn more. Now on to Anaerococcus vaginalis. As the name suggests, this one is found in the vaginal flora, right?
You got it. It's a common resident there.
Okay.
And it can cause gynecological infections. It's also been isolated from other sites, which suggests that it can be opportunistic and cause infections elsewhere in the body.
Interesting. What else should we know about Anaerococcus vaginalis?
Well, it's a butyrate-producing species, and it has a very distinctive PEP profile, so that actually makes it relatively easy to identify compared to some of its trickier relatives.
Well, that's always helpful. Clinically, what kinds of infections are we seeing it cause?
Well, it's been isolated from all sorts of infections, gynecological infections, superficial infections, even brain abscesses, so it can definitely cause serious problems. More research is needed to fully understand its pathogenic potential, but we shouldn't underestimate it.
Okay, good to know. Now we've covered the major players, but what about those less common GPAC genera, like Sarcina, Ruminococcus, and Cuprococcus? Do we need to worry about those?
These genera are really interesting because they highlight just how diverse the GPAC world is.
Okay.
Sarcina, for example, is unique because it forms spores.
Oh, wow.
Which is unusual for coccus. It's a strict anaerobe, needs carbs to grow, and it's often found in soil and the gut, especially in vegetarians. Clinically, it's pretty rare, but it has been associated with gastrointestinal infections.
So not your typical GPAC, but still capable of causing trouble. What about Ruminococcus and Cuprococcus?
Well, they're both strictly anaerobic and strongly fermentative. They rely on carbs for energy. They're major players in the gut flora, but because of their strict anaerobic needs, they're rarely isolated from clinical specimens. We're still figuring out exactly what role they play in human health and disease, but they're thought to be important for the gut microbiome and might contribute to certain conditions, like inflammatory bowel disease.
So they're probably out there causing more trouble than we realize, but just too hard to grow in the lab with our standard methods.
Exactly. And this brings us to two genera that aren't technically part of that clostridial group that most GPAC belong to, Slackia and Oedipobium.
Okay, tell me about these outliers. Why are they grouped with GPAC if they're not in the same family?
Well, they're not part of the Clostridiales order, but they do share some characteristics with GPAC, especially their morphology and the fact that they're anaerobic. Slackia exigua, for example, that one's a tiny coccus that's actually more closely related to bacteria in the Oedipobium genus than to other GPACs.
Ah,
It's strongly proteolytic, and it's often found in periodontal and periapical infections. It's also been isolated from deep organ abscesses.
so another potential troublemaker in the oral cavity. What about Oedipobium?
Oedipobium parvulum is the main species to be aware of. It's a strict anaerobe, produces lactic acid, and it's considered part of the normal gut flora. We sometimes see it in abdominal abscesses and wounds.
Got it. So even though they're not true GPAC, they can cause similar types of infections, and we need to be aware of them. Now, we can't forget about the gram-negative anaerobic Cocci. They often get overshadowed by their gram-positive counterparts, but they're still part of the normal human flora and can definitely cause infections.
Oh, absolutely. They're not as common in clinical infections, but we still need to pay attention to them.
All right. Let's shine a light on these often neglected microbes. What are the key players in this group, and what should we know about them?
The main genus to know is Veillonella. There are actually several different species, but they're really tough to tell apart based just on their phenotypic characteristics.
Okay.
They're part of the normal flora in the mouth and gut, and they've been linked to oral infections.
So identification can be tricky.
It can be, yeah. They don't always react well in those standard biochemical tests. Molecular methods like RFLP analysis of PCR-amplified 16S rRNA can be helpful for getting down to the species level.
Okay, good to know. And clinically, what types of infections do we typically see them causing?
They're most often associated with oral infections, but they can also show up in infections outside the mouth. Things like abscesses, pneumonia, and wound infections. Their lipopolysaccharide, LPS, is considered a potential virulence factor.
LPS, right? Remind me how that contributes to their ability to cause disease.
LPS, or endotoxin, is a component of the outer membrane of gram-negative bacteria, and when it's released, it can trigger this big inflammatory response in the host, leading to tissue damage and other complications.
Got it. So even though they're not as common as GPAC, they can still cause problems. What about treatment? What are our options for Veillonella infections?
Well, they're generally susceptible to metronidazole, beta-lactams, and clindamycin. But like always, susceptibility testing is recommended.
Okay, good to remember that. Any other gram-negative anaerobic cocci we should be aware of?
There are a couple others, like Acidomycoccus and Megasphaera, but they're very rarely isolated from human infections. Acidomycoccus fermentans, for instance, is found in the gut, and it uses amino acids for energy. While Megasphaera elsdeni is found in the stomachs of ruminants and occasionally in humans, they're generally not considered major pathogens, but it's good to know they exist.
So definitely not our everyday culprits. Well, it sounds like we've really explored this vast and intriguing world of anaerobic Cocci, both gram-positive and gram-negative.
I think we've hit the key points for medical microbiologists. It's a diverse group of organisms with a wide range of clinical significance, and it's essential to stay up to date on the latest classification, identification, and treatment strategies.
Yeah, it really is amazing how much more we know now about these organisms, especially with all the advances in molecular techniques. It's like we're finally seeing them for what they are, understanding how complex they are, and really appreciating that role they play in both health and disease.
Absolutely. It's a whole new way of thinking, moving away from those old broad classifications and really getting a much more detailed understanding of these organisms. And that's essential for accurate diagnosis and treatment.
For sure. I feel like I've gained a whole new appreciation for the world of anaerobic Cocci. But I bet our listeners are still processing all this information. What are some of the key takeaways you want to emphasize before we wrap up?
Well, I think the most important takeaway is that GPAC are clinically relevant, and we can't just dismiss them as harmless commensals.
Right.
They can cause a whole spectrum of infections, from minor skin infections to really serious life-threatening conditions, and we often underestimate their role.
We definitely need to take them seriously.
And we can't rely on those old assumptions about identifying them or treating them. Those traditional biochemical tests, they're just not enough anymore. We need to embrace the newer techniques like PEP kits and molecular methods so we can really get that accurate species-level identification.
Species-level ID is crucial, both for figuring out what role they play in disease and for picking the right antibiotic.
Absolutely. And remember, antibiotic susceptibility patterns can be completely different between different species, so that one-size-fits-all approach just won't cut it. We need to be precise and really target our treatment based on the specific bacteria causing the problem.
I think we've made that very clear. Any final thoughts for our listeners? Something to ponder as they go back to their labs and tackle the world of anaerobic Cocci?
Well, the world of anaerobic bacteriology is always evolving. We're discovering new species, antibiotic resistance patterns are changing all the time. Staying up to date on the latest research and guidelines, that's absolutely crucial for us as medical microbiologists.
A good reminder for all of us. And speaking of staying up to date, if you're looking for a great resource, I highly recommend checking out Principles and Practice of Clinical Bacteriology, 2nd edition. It's a fantastic resource for anyone working with anaerobic bacteria.
It's definitely a must-have for any microbiology lab.
Okay, time for one last challenge before we go. Let's test your knowledge with a few exam-style questions. You ready?
Bring it on. I'm ready for anything.
All right, question number 1. True or false? The term anaerobic streptococci is still an accurate and appropriate term to use.
False. That term is outdated and misleading. These organisms aren't related to Streptococci at all. They belong to a different branch of the bacterial family, and we should really use their proper genus and species names.
Excellent. Question number 2. Which GPAC species is particularly known for its ability to produce hydrogen sulfide, and how might that contribute to its ability to cause problems in the oral cavity?
That would be Micromonas micros. It loves to live in the mouth, and that hydrogen sulfide production, well, that probably plays a big role in how it causes tissue damage.
That's right. Okay, question 3. Let's say you get a sample from a diabetic foot ulcer. The gram stain shows gram-positive Cocci in clusters. What's at the top of your list of possible culprits, and what steps would you take to confirm the identification?
Well, considering the source and what we see on the gram stain, Phainogoldia magna would be my top suspect. It's a frequent cause of diabetic foot infections and often shows up as gram-positive Cocci in clusters. To be absolutely sure, I'd use a PEP for species-level identification and look at colonial morphology and other characteristics.
Sounds like a good plan. Combining those classic microbiology observations with the newer techniques is so important for accurate diagnosis. Now, for question number 4, can you explain why getting to the species level is so important when you're dealing with a GPAC infection?
It all comes down to choosing the right antibiotic. Different GPAC species can react in very different ways to antibiotics, so if you use a broad-spectrum antibiotic without knowing exactly which species you're dealing with, you might not be treating it effectively, and you could even be contributing to antibiotic resistance.
Okay, last question. A colleague comes to you with a question about a Streptococcus anaerobius isolate. They're not sure which antibiotic to choose because they've heard conflicting information about its susceptibility patterns. What advice would you give them?
I'd remind them that Streptococcus anaerobius can be a little tricky because it can develop resistance to certain antibiotics, especially some of those beta-lactams. I'd tell them to always check the specific susceptibility patterns for their isolate, Don't rely on general assumptions. Susceptibility testing is always the best way to go.
Excellent advice. It really shows how important personalized medicine is. Even in the world of microbiology, we can't just make assumptions. We need to tailor our approach to the specific patient and the specific microbe.
Absolutely. And that's where our expertise as medical microbiologists really comes in.
Well said. It's been a fantastic deep dive into the fascinating world of anaerobic copies. We've explored how they're classified, how they cause disease, the clinical features of those infections, how we diagnose them in the lab, where they hang out, and how we treat them.
We've covered a lot of ground. Hopefully our listeners are feeling more confident about identifying and managing these sometimes overlooked but definitely clinically important organisms.
I hope so too. And remember, the world of microbiology is constantly changing, so stay curious, keep learning, and never stop questioning. Thanks for joining us on this adventure. Until next time, keep those microbes in check.